thank you for joining me today, Michael.
Sure. Thanks so much for having me.
You are a researcher from the Department of Environment Sciences at the University of California, correct?
That's right. Yeah, I was a postdoctoral researcher there. I finished that position last year, and right now I'm teaching at a liberal arts school. We're teaching remotely because of the virus, but so this work was the work that we're going to talk about today was work that I did while I was a researcher there at University of California Riverside. That's
Okay,
right.
for sure. I guess we'll kind of just jump right into it. So can you tell me a bit about the research that you did both in California and then I guess specifically kind of connecting the Canadian light source as well?
Sure. So really the overall project for this started around 2015, and this is a time when California was just coming out of a really, or was still really in the midst of a pretty intense drought. Sort of at that point had been going on for about 4 years, and just to give a little bit of context, the drought to that point had cost about $800 million in lost crop revenues and about another $450 million in increased groundwater pumping. So the cost of just pumping groundwater to offset less surface water, less precipitation, added a major cost to the state. And so the overall project that I was a part of was kind of tasked with looking at how we might use active soil management methods or different types of irrigation methods to either reduce water use but provide the same amount of yield. And so that's really the project. And the part that I focused on was looking at how we might manage soil, in this case with cover crops, to increase the water holding capacity of soil. And so sort of how the study went about was looking at different two fields where they were split, and they were in a tomato corn rotation, and half of the field had cover crop and half didn't. And so by sampling those two fields and analyzing them, we were able to kind of track where carbon goes from the cover crop into soil, how that affects soil aggregation, and then and ultimately, we think, based on some other research, that this is sort of the mechanism that leads to increased water holding capacity in soil. So having larger aggregates or these sort of balls of things that are stuck together in soil can really increase the amount of water that can be held in the soil. This is something tying back to, say, in a drought situation, that would be really advantageous if you could store more water from a wetter year in soil. And that would be something that could help mitigate the effects of drought. The way that the Canadian light source really helped and was essential to this research was at the Canadian light source, there's a beam line, the SGM beam line that we use, that allows us to look at the chemistry, really the chemical composition of carbon in different components of the soil. And this is pretty unique just to CLS that they have this capability. They're one of only a few places in the world that we would be able to look at the carbon chemistry of different components of the soil this way. And so we're really fortunate to be able to work there and use that facility as part of our research.
For sure. So then when exactly were you doing that work at the CLS?
So I've been working at the CLS on other projects as well as this one since about 2013. And then the work for this project was conducted around 2017, 2018 was the time that I was really up at CLS a few different times working with these samples that were part of this study specifically. Yeah.
For sure. And so obviously the goal of the study was just kind of to see what cover crops could do for the soil, correct?
That's right. Yeah. And so there have been a lot of studies in the past that look at different properties of, say, how large aggregates become when you have cover crop compared to not having cover crop. One of the real motivations for us was to really understand where the carbon from say, the cover crop roots or the cover crop plant material goes in different components of the soil. And so we use a couple different ways of assessing soil aggregates. We look at them both with size and then with how dense they are. And this allows us to kind of create a sort of conceptual model of how aggregates are constructed with carbon. Really, you can think of as sort of the glue that holds these balls or aggregates of soil together. And so we were really interested in what the chemical composition of that is. Is there a certain type of carbon that the cover crops are putting into soil that can really help form these kinds of aggregates? And so we kind of took a more chemical approach to looking at this sort of phenomenon of increasing aggregation from cover crops. That's really kind of the part that we were trying to add to the research on cover crops in general.
For sure. So then I guess basically, what were your results? What did you guys find from this study?
So this study was done over a short time period. It was done over a single season of cover crop, and there's a couple important. That's generally not how these studies are done. They're generally done for 5 years, maybe longer, sometimes much longer, to see if you're building an increase in total carbon in the soil. What we wanted to look at is over just a really short time period. We didn't really expect to see an increase in the total amount of carbon, and we didn't. We found that over just 1 year, we didn't increase the total amount of carbon with cover crop. But what we did find is that the carbon that's coming from cover crop, and we can assess that both in terms of the carbon coming directly from a plant has a little bit different chemical composition, and it also has a different signature that we can measure using carbon isotopes. And so we did that as well. And so we can tell that there's carbon coming from the cover crop, and then it's moving into aggregates very quickly over the course of just 1 year. And so what that means is that the total amount of carbon doesn't increase over a year, but we do see that there's new carbon from the plants that are from cover crop, and that's getting into these soil aggregates very quickly. And so that kind of result is really a main finding of our study. We also look a little bit at the changes that cover crops have on the minerals in the soil, and so if cover crop is changing the properties of different minerals in the soil, that can also have an effect on how much carbon and water can be retained in the soil as well. Yeah.
For sure. And so then the work specifically that you did at the CLS, like that was able to tell you more about that carbon that was in the soils. Can you maybe talk a bit more about that?
Sure, that's right. So when we sample the soils, we'll have a sample of, we call it a bulk soil. So it's just the soil. And then what we might do is we might separate it into different sizes using sieves. We do this while the soil's wet. We might also separate it into different densities. And we think of this as things that are mostly mineral. These would be the more dense fraction of your soil. It's mostly things like clays, oxides, sand, things like that. And then as you get less and less dense, in terms of the part of the soil that's less dense, that's more like your pieces of dead plant matter. These things are less dense than the minerals in the soil. And so if we separate those out, and then we can take those sort of fractions of the soil and analyze them at CLS to see which part of the soil has more of organic matter or carbon that's coming directly from the plants. And then we think of that in terms of those pieces that we've separated the bulk soil out into are kind of like the building blocks of the overall soil. And so we can look at, we can kind of take the soil apart in this way, analyze the different components, and then think about how that might be built back up in sort of the bulk soil, so we can track what part of the soil the carbon from cover crops is going into. And that's really, to be able to do that, we need to work at CLS to be able to analyze, once we've separated the soil into all these different pieces, we need to be able to analyze what the carbon chemistry is like in each of those pieces. And so that's really where we use CLS.
Awesome. Well, I just want to also open it up to you if there's anything else you want to add on this topic that maybe we didn't touch on yet.
Let's see. So I would say, I think I touched on, I wanted to just stress the kind of the long-term sort of benefits that we've seen of cover crop from a lot of other studies. But other than that, I think you'd ask, and you're based in Canada, right? And so one of the things when we apply to work at the Canadian Light Source is they ask, what's the benefit to Canada? And so coming from California, it may not seem obvious, but when I looked into it a little bit more, there were, Canada actually imports a lot of produce and agricultural products from California. And so one of the motivations in terms of continuing to work at CLS is that any improvements that we make in California agriculture directly tie to prices and to the sort of availability of produce that California exports everywhere, including Canada. And I think that that if you're thinking, okay, well, these guys are doing this research over in California, how might that apply to us? I think that we try to focus on sort of understanding at a sort of fundamental level what's happening in soil with cover crop, and the idea with that being that can hopefully be generally applied to different types of soil, not all soils, but hopefully somewhere more than just a specific location. And then that agriculture is tied to all different places, and especially in a place like California where we're a large agricultural exporting state. And so those are a couple of the other things I would just add in general terms. So yeah.
Awesome. Well, thank you so much for chatting with me today, Michael. I really appreciate it.
Sure. Thank you so much. And I hope you're staying safe and healthy during all this time.
Yeah, for sure. You as well.